Hi, I'm Philip Stevens, CEO and co-founder of Noscendo, and welcome to this DISQVER introduction. DISQVER is our digital pathogen identification platform, which identifies over 16,000 microbes and has the ability to detect over 1,500 pathogens. Those pathogens can be bacteria, DNA viruses, fungi, or parasites, and we are able to detect it just from a single blood draw of a patient. Even if your patient has received or is under active antimicrobial therapy, DISQVER remains highly sensitive and highly specific. Unlike PCR or blood culture, therapy doesn't have an impact on DISQVER sensitivity. Using next-generation sequencing and cell-free DNA, we are able to deliver DISQVER results within 24 hours after sample receipt in any laboratory, giving the clinician the opportunity to treat their patients directed and specifically towards the identified pathogen just from a single blood draw. Unlike PCR, we test for 16,000 microbes simultaneously and not for a defined set of microbes like a PCR.
Diagnosis with DISQVER starts with a simple standard blood sample, drawn into tubes which we provide to you. This can be venous or arterial blood. The blood sample is then transported either to a Noscendo lab or to a certified DISQVER provider, which is a laboratory near you that has been certified by us. In the lab, the blood sample goes through a number of preparation steps, is then sequenced using a high-throughput sequencer. The result is a complete analysis of all cell-free DNA in the blood sample. This includes all of the microbes in the patient sample. Then this data is uploaded to the DISQVER platform. In the first step, DISQVER removes all of the patient's human DNA from the overall analysis. Technically, this is done using algorithms developed by Noscendo. In the second step, DISQVER matches the remaining DNA with a large reference database. It contains information on the DNA of more than 16,000 microbes, including more than 1,500 bacteria, fungi, DNA viruses, and parasites. Since the cell-free DNA of pathogens is still detectable after the administration of anti-infectives, the results are not effective by any anti-infective treatment. In the third step, DISQVER analyzes the quantity and relevance of previously determined microbes. And yes, finally, a digital report is generated that summarizes all of the results in a clear and concise manner. The entire process takes less than 24 hours from the receipt of the sample in the lab. The report is transmitted digitally to the clinic or lab. It contains an evaluation of all relevant bacteria, fungi, DNA viruses, and parasites detected in the patient sample. Thus, DISQVER provides precise info that can help you to optimize the management of your patient and decide, for example, which is the most effective treatment for the pathogens that have been identified. How does DISQVER help clinics, physicians, and patients? DISQVER determines more than 16,000 microbes in just one blood sample, including over 1,500 bacteria, fungi, DNA viruses, and parasites, even after anti-infective therapy. Atypical pathogens are identified on the first run. Thus, DISQVER is hypothesis-free. No assumptions need to be made before the test is run on what type of pathogens may be present. Since there's no need to wait for the cultivation, the result is available after less than 24 hours from receipt in the laboratory. DISQVER also offers doctors the opportunity to rule out possible infections before major treatments or interventions. The precision of DISQVER provides physicians with detailed information on the pathogens in the blood and can thus optimize the treatment of seriously ill patients.